Good morning, everyone. Good to see everybody. I hope everybody's doing well today. Yes, please go ahead and type your first name into chat for me. So I have a record that you came this morning. Thank you. Very good. Good to see you guys. I missed you on Monday. I know you were busy taking your first lab practical, but I missed seeing your smiling faces. Yes, and hopefully you saw my announcement that the lab practicals are graded, so you can go and look not only at your grade, but you can look at my comments. Always take that opportunity. Don't just look at your grade and think, oh, that's great, I got an A. Open up your exam and click on where it says view comments or view instructor comments, and take that opportunity, because sometimes there may be just a couple of words that I've left for you that might be helpful to you moving forward. I always go through examinations by hand. Obviously, for the lab practicals, all of the questions have to be hand graded. But even the lecture exams, I do go through by hand, because sometimes there are comments I want to leave for you to help you maybe understand why you got something wrong, or what I was looking for in an answer. So that's always a good idea. All right. Looks like just about everybody's here now. Missing a couple folks. Very good. So today is your second lecture exam. I saw that a couple of you have already taken the exam early this morning. Good for you. Good for you for getting up and getting that taken care of. So just be sure to get that lecture exam into me by 8 o'clock tonight. Remember, you're free to use your notes, your lecture notes, during that exam. You're just not allowed to use any other source material. So you're not allowed to Google any questions or get help from anyone else. You have to rely on those lecture notes that you took while you were watching the lectures. Very good. All right. So our work for this morning is a laboratory exercise called Microbes in the Environment. And we are moving forward now out of the first laboratory unit, which was really about introducing us to the world of microbes and learning how to name the parts of the microscope and how to focus the microscope and things like that. And of course, the basics of examining microbes under a microscope. Remember, we talked about examining living microbes versus examining stained microbes. And we talked about simple staining and differential staining. And we even looked at one of the differential staining procedures. We looked at gram staining, which is the single most common staining procedure that is used in microbiology. So now we're ready to move forward into the second unit. Now we have a lot of information under our belts. We have a lot of new vocabulary under our belts, hopefully. And we're ready to move forward into a little bit more complex laboratory topics, including what we'll be talking about today. looking ahead to next week, we're moving on not only in laboratory, but we're moving on to a new unit in lecture as well. And I will give you a fair warning that we're moving into more difficult lecture material now. We've really gone through the basic sort of review material that was really in the first lecture unit. And then we moved into some more detailed review, which was the second unit. And now we're moving into really some more of the meat of the course. Next week, for example, you'll be viewing a lecture called Biochemistry. That lecture is one of the more difficult ones that we do in this class. Now, we all come from different backgrounds. And yes, you've all had certain prerequisite courses, but before those courses, you know, your background was very different from one of your classmates' background. That's just the way it is. So this biochemistry material might be review for some of you, and it might be really quite new for others. It's a single lecture, so we're not going to learn all of biochemistry, but we're going to review the things in biochemistry that are especially relevant to microbiology. So some students don't enjoy chemistry, and that's fine. That's fine. No judgment here. But if you're one of the people that doesn't really enjoy it, maybe you find it particularly challenging, just be sure to give yourself the time to watch the biochemistry lecture, you know, at a comfortable pace for you so that you can take as many notes as you need to take in order to have that material under your belt. So I'll try to warn you when you have the more challenging lectures coming up in your schedule. So let's pull up Canvas now and take a look at what we have on our schedule for next week. Let me get rid of this. And that's what you should be seeing on your screen right now. You should be seeing our modules page. Here's this week, the lab practical exam, the lecture exam. This is looking ahead to next week. So here's that biochemistry, at least the slide set for the biochemistry lecture on YouTube. Here's the quiz. The other lecture topic next week is microbial growth. Now remember, when we talk about growth as it relates to bacteria and other microbes, we're talking about cells increasing in number. So unlike, say, a human child that grows and that single organism gets bigger and bigger and bigger, when you talk about single-celled organisms like bacteria and other microbes, growth is about the population growing. It's about more and more cells being produced through cell division. And that's what that lecture talks about. It talks about this process that particularly bacteria use to increase their cell numbers. In laboratory next week, we'll be talking about the fungi, and we'll also be looking at a special category of culture media, which is used to help us identify the genus and the species of the kinds of microbes that arrive in the microbiology lab. Remember, a lot of what we do in microbiology, at least as it relates to healthcare, a lot of what we do is we identify microbes. We identify the genus and the species of the microbe that arrives in the lab. And as you know, you can't just look at a bacterial cell and know what it is. You cannot identify its genus and species just by looking at it. We have to do a number of different tests on that cell in order to know exactly what it is. And we start off by doing things like gram staining, because that's going to help us in that identification process. But then we also have to use other tests in order to see what characteristics, what traits these microbes have, because that will help us identify them and report back to whoever sent that sample to the lab. What we're going to be talking about today, though, is not related directly to a healthcare and to a patient, but rather to the environment. Because the other thing that happens in microbiology labs is samples get taken out in the environment, and those get sent to the laboratory again, so we can identify the microbes that are in those samples. So we're going to receive samples from lakes and streams and ponds, and we're going to receive soil samples, and we're going to receive samples from buildings, commercial buildings. We receive food items, perhaps that are suspected of having caused some food poisoning. We have to identify the microbe that is in that sample. We can't just look at them and know. We have to do a whole lot of testing in order to determine what microbe we're dealing with. So today we're going to focus not on patient samples, but environmental samples. We're also going to talk in a little more detail about culture media. We're going to start increasing our vocabulary around these different materials that we use in the lab in order to grow microbes, because there are so many different types of microbes and they all have different requirements in terms of what nutrients they need and what temperature they grow best at, and so on and so on. So that'll be our focus today. All right, looks like everybody's here, which is wonderful. I would ask, moving forward, just as a reminder, that you go ahead and you leave your camera on, your video camera on during the laboratory, and you mute yourself unless you're speaking. If you need to step away, feel free. You can either just step away from your camera, or if you would rather, you can turn off your camera while you step away. But if you have to step away from your camera for some period of time, go ahead and send me a private message in chat and just let me know, so I'm not looking for you during the laboratory. Believe me, I understand that, we have to get up. Sometimes we have to go to the restroom. Sometimes we have to refill our coffee cup. I get it. But yeah, just let me know what's going on with you. Okay, very good. So I'm going to go ahead and pull up our slide set for this morning. And that's what you should be seeing right now. You should be seeing the title slide for microbes in the environment. So these are our objectives for the exercise this morning. The first thing we're going to talk about are these environmental samples. We're going to talk about how we collect samples from the environment, generally speaking, and some of the testing methods that we use when we're trying to figure out what kind of microbes are in the environment around us. Because of course, there are always microbes, but sometimes there are pathogens. There are microbes that can cause disease, and we have to keep track. We have to monitor so that we know that the environments that we're in every day are safe. Now, in terms of culture media, we're going to talk about why agar, which is the material that we use to make solid culture media, why agar is such a good choice for that. We're going to compare the way that we prepare agar in the lab. We'll talk a little bit about plates and slants and deeps. Those are all different kinds of agar containers. We'll also take a look at the difference in growth patterns of bacteria on solid versus liquid media. Because of course, cells are going to look different to our eyes, not under the microscope, but just our naked eyes. Cells are going to look very different when they're growing on a solid surface, like an agar plate, versus when they're growing in a broth tube. The last thing we'll talk about today is a little bit about how we look at cells growing on an agar plate and determine what's called colony morphology. I'm not even going to try to explain this right now. We'll wait till we get there. Okay, so as I said a minute ago, microbes are everywhere. We have to keep reminding ourselves of that. Microbes are everywhere. They are on and in all living organisms, including you and I. Don't forget your microbiome. They are all over our natural environment, and they are all over our man-made environment. In other words, they're all over our commercial buildings and our places of work and the businesses that we frequent, and so on and so on. And just like we always have to remind ourselves that microbes are everywhere, we also need to remind ourselves that most microbes are completely harmless. In fact, many of them are beneficial, as we talked about, but a few of them are pathogens, and it's helpful to us, the folks who are involved in public health and other health care related fields. It's helpful to us to monitor those environments for the presence of pathogens. So hopefully we can prevent disease. There are several different types of environments that we routinely monitor. We live in a state that has an economy that is largely based on tourism. And we are in the time of year right now where we want lots and lots of tourists coming and enjoying our hiking trails and our beaches and our lakes and rivers and ponds. So recreational water bodies are a big, big place of monitoring for us here in New Hampshire. We have to keep track of the cleanliness of the water in terms of, again, pathogens. We also have to monitor water quality at water treatment plants, of course, because we want to make sure that water has been sufficiently cleaned up at our water treatment plants. Public water systems, the supply that comes into many of our homes, those have to be monitored. We also have to monitor buildings. We have to monitor hospitals, of course. We have to monitor medical offices. We have to monitor care facilities. We all got a hard lesson in the importance of monitoring for microbes in long-term care facilities during this pandemic. We got a very hard lesson in how easy it is for certain pathogens to be transmitted in long-term care facilities. We also have to monitor places that you might not think of at first, places like industrial clean spaces. If you're not familiar with that terminology, there are industrial processes that require monitoring because these spaces require no particulate matter being present. So that means no living cells and no dust particles and nothing can be in the air in these places because they are manufacturing something that needs that level of cleanliness. So if you think about, for example, pharmaceutical manufacturing, if you think about electronics manufacturing, the folks that make the chips that go into our devices, those places have clean rooms. They're called clean rooms. and they have to be monitored so that they know that there's nothing microscopic in that room that might interfere with the production of their product. Okay? So, lots of places that need monitoring. And this is one of the jobs, sorry about that. This is one of the jobs that we do in microbiology. Now, there are lots of different ways to sample our environments, depending, of course, on what you're sampling. We can sample surfaces, and we can use that sample then to inoculate culture media and see what grows. So very, very common to sample solid or semi-solid surfaces, things like drains, floor drains, and refrigerator units and vents. Look at that dirty vent. Just looking at that picture gives me all kinds of anxiety. We can monitor these surfaces just by collecting a sample, by taking something like a sterile swab, which is what you're looking at here, and rubbing it across the surface of this material that we want to sample. We can filter air. So in commercial spaces, in buildings, we often do this. We can put a piece of filter paper into an air vent and allow air to pass through it. That will trap microbes in the filter. And we can use that piece of filter then to inoculate media. And of course, we can also collect liquid samples. Remember, if we're collecting a sample of water from a natural environmental source, or even a place like a water treatment facility or a municipal water supply facility, the sample that we are collecting is most likely too dilute for us to use directly. So we usually centrifuge those samples so that we can concentrate the cells down and then inoculate media in the lab. We take this sample into the laboratory, but we often are not able to use it directly. In other words, when that swab comes in, or when that bottle of water comes in, we're not able to use it right then and there. We often have to take some cells from that sample, or take that sample and put it on some kind of culture media, and then encourage cells to grow. We need as many cells as we can get, in other words, in order to determine what kind of cells they are. Okay? All right. Now, environmental samples that are collected with these sterile swabs, and that's what you looked at in this picture. This is a commercially available device. You buy these, they come sterile. You can see that there's a cap at one end because this thing comes with a tube. It comes inside a tube and it's sterile so that when you take it out of the tube, you can get your sample and then you can put it right back into that tube and send it to the laboratory. Now, these transport tubes, they're not empty. They have a little bit of culture media in them. You're looking at a couple of different transport specimen containers here that, again, are available to us commercially. We buy these. You can see that there are different colored handles on each one of these. Those are color coded according to what kind of specimen you're collecting. When you take this swab, and I know it's hard to see on your monitor, but there's a swab coming off of that red orange handle. When you take this and you rub it across the surface of something that you want to examine, and you put it back into this transport tube, there's a little bit of media in here. And we call this media transport media. I have it written down here at the bottom of the slide for you. What transport media is designed to do is keep the cells that might be on this swab. It's It's going to keep those cells alive and happy until they get to the lab. There was an extra credit, a bonus question that you all saw about a sample that was collected from a patient and then sent to a laboratory, but it took 2 days for it to arrive. And you were asked in that question to think about what kind of problem that might cause and why you might not get a good smear made from that sample. Well, now we can talk about transport media. Transport media is not designed, and this is important, it's not designed to encourage those cells to grow. A lot of the culture media that we use in the laboratory is designed to help cells grow. We put the cells into the media, we put the container into the incubator, and then we wait because cells are going to start growing in there. They're going to start dividing, multiplying. Transport media is not like that. Transport media is very minimal. It has just enough nutrients in it to keep those cells alive without encouraging them to grow. We don't need them to grow. We just need them to get to the lab so that they can be then identified through different processes. So these transport media containers are really important. We use them both for patient samples and for environmental samples. So if a physician, for example, takes a swab of the back of your throat because they think maybe you have a bacterial infection in your throat, especially if if they're going to send that sample to the microbiology lab for testing, that then has to go into some transport media, and then it will be sent to the laboratory. Someone will come from Quest or LabCorp, and they will pick up those transport containers every day, take them to the laboratory where they will be worked with for diagnostic purposes. Now, obviously, there are times when we're not using a swab to sample a solid surface, but instead we're collecting water samples. Like I said, this is a very common activity in the summer in New Hampshire. What you're looking at here is a lake where people are allowed to swim. And you can just see this buoy line right here. That's cordoning off where the swimming area is. That's in the foreground of the slide. And you can see two young ladies in their waders out in the water, and they are collecting samples for analysis. So we have here in New Hampshire, we have a Department of Environmental Services that does this kind of work. They send people out to recreational swimming areas and they gather samples all summer long to monitor for different pathogens, because we don't want people swimming in this little area if the water is not of a high enough quality to swim safely. Now, fresh water is commonly sampled, but so is ocean water, and that's what you're looking at here. There's someone collecting ocean samples here, again, in the area of the beach. Here's someone who is collecting from water that no one is swimming in, right? But this might be a fishing hole. This might be a place where people tend to fish. Or this might be a place where a lot of wildlife comes. And so this water body is being monitored as well. Notice that the person is not in this water. They are using this very handy telescoping pole with a collection bottle at the end, so that they don't have to actually get into this murky brown looking water. Of course, we also monitor well water. It's very, very common here in New Hampshire for people to live in homes that are not connected to the municipal water supply. Instead, they have their own private well. And now the state doesn't monitor private wells, but the homeowner can do it. The homeowner can request that water samples be collected and analyzed to make sure, again, that the water is safe for drinking. You'll notice that the bottles that are used for this kind of work are just basic plastic bottles. Again, you buy these commercially. There are different types of plastic that are available, different sizes are available. It all depends on your needs, what kind of sample you need to collect. Now, one of the most important things to know about water sampling is this. You cannot go into a swimming area, for example, like this one, and take a single sample. You need to collect enough water so that you have a representative sample from this swimming area. So you're going to need to collect some water from up close to the sand. And you're going to need to collect some from the deeper areas of the swimming area. You're going to want to take samples on the right and samples on the left and some in the middle. You want to get enough of a sample so that the whole swimming area is being tested. Same thing down here on the beach. You would not take a single sample on a beach where people swim. You would want to get multiple samples so that you felt that you were getting a real representation from that area. So we don't need to put water samples into transport media because they're already in liquid. They're already in water where they normally live. If there are any microbes in that water, that's their natural environment. So we don't need to put them into any special container. They are just gathered up into these plastic bottles and then sent to the lab. But remember, if you're gathering water from a lake or a river or whatever, that's going to be a dilute sample. When you take a swab and you swab it across a solid surface, and then you put that into transport media, that's not going to be very dilute. But when you gather water, that's going to be dilute. So the water samples have to be centrifuged in order to gather all those cells down to the bottom of the container. And then we can take some of those cells and use them to create a culture in the laboratory. Okay. So we can sample solid surfaces. We can sample water. We can also sample the air, as I mentioned. And if you remember, I said that it's possible to use filter paper and allow air to pass through that filter paper. We do that in ventilation systems a lot. But I also wanted to show you these devices. These are commercially available monitors. They are used to measure not the cells that are in the air, but spores that are in the air. There's a real growing industry in residential homes for people to hire companies to come in and monitor the air quality in terms of microbial spores. And what these devices do is they essentially pull in an air sample into the device, and then they give you a readout on how many spores are being detected. If you go onto the internet, you can find these companies. They're all over the place, and they will tell you, oh, you should hire us because we're going to come in and tell you whether the air you're breathing is safe. Okay, here's the problem that you should know about this kind of environmental sampling. Those devices, they're going to tell you how many spores are in the air. What they're not going to be able to tell you is whether they are bacterial spores or fungal spores, and whether they're pathogenic. We live in New England, and we live for a large part in old houses, right? I don't know if you own your home or if you rent, but there are lots and lots of houses in the New England area in New Hampshire that are old. right? Couple hundred years old, some of them. And we love our old houses here. And I can tell you that old houses are full of spores. Remember why things sporulate. Bacteria sporulates because the environment is not good for it to grow in. So bacteria sporulate when the environment is too dry or there's not enough nutrients, and they simply cannot survive in a cellular form. There are lots of different bacteria that can sporulate. There are two types of bacteria, two genera of bacteria that are pathogens that can sporulate, Bacillus and Clostridium. Those devices won't tell you whether you have the dangerous spores. They'll just tell you if you have spores. And yes, you have spores in your house. You do. But the chances are very good that they're not the kind of spores that can harm you. Remember, most spore-forming bacteria come from the soil, and they're not harmful to us. So it's unfortunate, but there are some less than scrupulous companies that will come into your home and they will tell you, oh, you have 20,000 spores per cubic so-and-so of your air, and isn't that terrible and dangerous? And you should let us come in and clean your air. That's bad practice because you're not in danger in that situation. And they may be able to filter your air out for you, but it's just gonna go right back to what it was after they leave. Now, the other thing to know, we haven't talked about the fungi yet, but most spores that are in homes are fungal spores. And those are not the same as bacterial spores. A lot of fungi make spores as reproductive structures. In other words, it's their version of sperm and egg. So reproductive spores are absolutely not harmful to us. And reproductive spores are all over the place. They're in your home, they're in your backyard, they're in your car, they're on your clothing. The fungi are everywhere. And yes, there are lots of fungal spores floating around, but they are not harmful to us. So, just something to know, that just because you have spores floating around in your home, that doesn't mean you need to do anything about it. That doesn't mean you're in any kind of danger. Now I want to talk just a little bit about this device right here. This is called a Rodak plate. And Rodak is an acronym. It stands for replicate, organism, detection, and counting. These are really interesting little commercially available devices that we use particularly in healthcare settings to help us monitor for microbes. And as you can see in this picture, someone is taking this Rodak plate and pressing it down onto some solid surface. Maybe it's a desktop at a nurse's station, or maybe we're in an operating room, or we're in a patient room somewhere. What a Rodak plate is something that looks very similar to a Petri dish, the plastic Petri dishes that we use in the microbiology lab. It even has this agar material. That's what the beige material is. But Rodak plates are not the same as plastic Petri dishes. First of all, they're very flexible. They're very bendable. And rather than being designed for us to place cells on, they are designed to be pressed down onto surfaces. So essentially, this plate is upside down. The agar is exposed on the bottom. This person is pressing their finger onto the plastic. And the open agar, the face of the agar, is on the bottom. So she is pressing the agar down onto this surface. She's trying to pick up any cells that might be on that surface. So you take this plate and you take the cap off, you flip it upside down and you press it onto surfaces. And again, the plate is very flexible, so it bends really easily. You just touch it. You touch it to surfaces. If there are any cells there, they're going to get stuck to the agar. And then you just tip the plate back over, put the cover back on, and you're done. Rodak plates are available commercially, which is nice. And of course, there are people in especially healthcare settings whose job it is, part of their job is to take samples every month. So they go around wherever they're assigned. Again, if it's a hospital, maybe they have to sample places in the OR, or maybe they're sampling places at the nurse's station or wherever, in the patient room, in a procedure room. And those samples are used to monitor what's going on in terms of microbes. Obviously, we want our hospitals to be as clean as they can be. But as you probably already know, hospitals are a place where a lot of bad microbes can be found. And that's because sick people come to hospitals. Sick people come into hospitals and spread microbes. So hospitals work very, very hard to try to keep track of what's growing in the building, in the environment, because they want to get rid of it. They want to disinfect and clean. It's very hard. It's very hard. But we work hard at it by doing this kind of monitoring. Now, what's interesting about Rodak plates, unlike the other samples we've been talking about, the Rodak plate isn't intended to tell us what microbe is growing. The Rodak plate is there to tell us how many microbes are growing. So what's going to happen on that plate that gets pressed down onto surfaces? That plate is going to go into an incubator, and we're going to allow those cells to grow. And then we're going to count how many little clumps of cells we can see on the plate the next day. Those are called colonies. Those are called colonies. And by counting colonies on these Rodak plates, we can get a decent idea of how many microbes there are in the environment. We don't know what they are. We don't know if they're dangerous pathogens or not. It just tells us how many there are, relatively speaking. And that's good information for a hospital, because there are some places in the hospital where you don't want microbes growing, right? Even if they're harmless to you, you just don't want them because they could be dangerous. We want our operating rooms to be very, very clean, right? So a lot of hospitals and other healthcare facilities use this Rodak system in order to just routinely monitor for how many microbes are present in the environment. Now, this is a commercial system. So it's, you have to purchase the Rodak plates and so on, but you don't have to use a Rodak plate to do this kind of monitoring. You can also collect your own samples with, again, swabs, sterile swabs, and streak Agar plates. And that's what you're looking at in this picture. So unlike this plate, which is this commercial Rodak plate, these are just plastic Petri dishes. And they have some of this solid growth medium on the bottom that we call agar, or nutrient agar. And what you can see in this picture is that something is growing on this plate. Something's growing here. These little sort of opaque cream colored circles are colonies. These are colonies of bacteria. Now up here, you can see more streaks of material growing. There are many, 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 many cells in a streak like this. But down here, we've managed to spread the cells out so that we get individual colonies growing. The other kind of agar plate that we like to create is this one. This one only shows colonies, and the colonies are all really nicely spread out across the plate. These two types of agar plates, these two plates serve very different purposes in microbiology. This plate over here is called a quadrant streak plate. And I have the term written right down here at the bottom for you. A quadrant streak plate is used to isolate colonies. So we are trying to create colonies when we make an agar streak plate. And the reason that we would want to make colonies is so we can examine them and determine what's called colony morphology. Morphology, if you break that word down, morphology means body form or the study of body form. When you put a cell onto an agar plate, when you put a bacterial cell onto an agar plate and you put it in the incubator, what's going to happen overnight is that cell is going to divide and divide and divide and divide and divide. And by morning, or by 24 hours later, I should say, you're going to have a visible colony of cells. They all came from that one cell. They're all identical. Every cell in that colony came from the same parent cell, and every one of them is genetically identical. They're clones. And that's because bacteria reproduce asexually. They simply divide themselves in half. So unlike you and I, who came from a mother and a father, bacteria produce their offspring by simply dividing themselves. And that's what a colony is. It's hundreds and hundreds and hundreds and even thousands of copies of that original cell. And it turns out that each species of bacteria produces a unique colony. It looks unique. And it just has to do with their species. Some colonies are very small, some colonies are very big. Some colonies have perfectly round edges. Some colonies have really irregular edges. We can look at a colony with our naked eye. Sometimes we also put them under something like a dissecting microscope, a less powerful microscope, just so we can see them a little more clearly. I like to use a dissecting microscope because my eyes aren't the best. But we can look at that colony and we can describe what it looks like. And that can be one of the ways that we work to identify a bacterium, by how its colony looks, by what its colony morphology looks like. Some bacteria have really unique colony morphologies, and I'll show you some pictures in just a bit. But some bacteria have very boring colony morphologies. A lot of different species of bacteria grow as circular, beige, or cream-colored colonies. Not very exciting. But sometimes colony morphology can help us identify a bacterium. That's why we make quadrant streak plates. We do it so that we can grow colonies that we can examine for colony morphology. Now, the other kind of plate I showed you is called a spread plate. And that plate is made for a whole different reason. Spread plates, like the name suggests, Spread the cells out all over the surface of the agar, nice and evenly, so that we get colonies all over the agar. We make spread plates so that we can count the colonies. So the quadrant streak is so we can examine them. The spread plate is so we can count them. Now, why would we want to count? Because if we are trying to determine how many bacteria are in that recreational lake, or how many bacteria are on that desktop, that we're afraid is contaminated with microbes. If we're trying to figure that out, we need to know how many bacteria per unit of surface area or water volume. We have to have a way to mathematically quantify microbes sometimes. We know, for example, out in a body of water, that it's okay if there's very few dangerous pathogenic E. coli in that lake. But if there's a lot of that pathogenic E. coli in that swimming area, we want to close that area down for a while. So we have to have a way to count, to quantify the cells that we grow from environmental samples. And that's what we use those spread plates for. Now take a look at this image here. This is a diagram. I know it's hard to see from where you're sitting, but this is a diagram that shows how we make these plates. And I just mentioned this when we were talking about our tools. If you start over here on the right-hand side, here's an agar plate. And what has happened in this plate is a little volume of material has been placed in the center. Okay. Now they're going to take one of these hockey stick tools that I mentioned to you, and they're going to use that hockey stick tool to spread that sample material all over that plate. This might be a sample that came from a lake or from a water treatment plant or whatever. Or it might also be something that we're growing in the laboratory. Maybe someone sent us a sample and we used that sample to inoculate a broth culture. And the next day, we're going to go ahead and make a spread plate. We take a little volume, we put it in the middle of the plate, we spread it around with the hockey stick, and then the plate goes in the incubator and we let the colonies grow. Now, there is a variation on spread plate called pour plate, P-O-U-R. Remember, there are some microbes, especially out in the environment, that have different metabolic requirements, especially in terms of things like oxygen. Lots of microbes grow very well in open air, but some microbes have difficulty growing in the presence of oxygen. So we also have a variation on spread plating called pour plating. And this time what we do is we spread the sample material, again, shown in pink. We spread the sample material into an empty dish. We pour agar over the cells and let it solidify. And then we grow them up. So the difference between a spread plate and a pour plate, here we are pouring the agar, is that these colonies are growing on the surface of the agar, and these colonies are growing in or under the agar. So again, the idea is the same. We make spread plates and pour plates because we want to count colonies. And we want to count colonies because we want to calculate how many microbial cells there are per unit of surface area, or per unit of volume, per unit of air. wherever we got that sample from. It doesn't help us identify the bacterium. I'm not going to be able to look at my spread plate and tell you what species it is. But I can look at my spread plate and tell you, you've got 1.5 million bacterial cells per cubic foot of air in your building. Okay, see the difference there? There are things we can do to identify bacteria, yes. But there are also things we can do just to count bacteria. So we have an idea, again, of how many microbes are in that space, in that environment. Now, one of the things to know about these spread plates or even the pour plates, what you're looking at here in this slide is a set of these plates. And of course, they look very different, don't they? This plate, it's hard to know what's happening on this plate. This plate is covered in so many colonies that they've all merged together. Okay, this is what we call a lawn of bacteria. Just like your backyard is covered in a lawn of grass. This is what we call a lawn of bacteria. There's no way that I could count the colonies on this plate. I couldn't do it. They're all merged together. The difference between this plate and each one of these plates is that I am slowly diluting the original sample before I put it on the agar. So I'm getting fewer and fewer and fewer colonies each time. So I take whatever material arrived in the lab and I do what we call a serial dilution, a series of dilutions. And I have that term right down here at the bottom for you. Serial dilution, step by step, usually by a factor of 10 each time. So this might be just the original sample that I got in the lab, and I just plated it as is. Then I took some of that sample and I diluted it 1 to 10, and I made this plate. And then I took this and I diluted it again, 1 to 10, and on and on and on. Now, why am I doing that? I'm doing that because I want to create a plate that I can count the colonies on. I don't want to try to count these colonies. It's just too hard. There's too many colonies on this plate. This plate right here has too few colonies on it. This plate is so diluted. I have now diluted the sample to the point where I've only got 2 colonies growing. I don't feel good about this sample either. I want to create enough dilutions so that I can choose the plate that has between 30 and 300 colonies on it. That number's right down here for you. I want to create a series of dilutions from my original sample, and I'm going to place each dilution onto its own plate. I'm going to put them all in the incubator. I'm going to come back 24 hours later. I'm going to choose the plate that has between 30 and 300 colonies on it for my counting purposes. So just by looking at this one, it's probably this plate. That's considered enough colonies to be accurate without being so many colonies that I have trouble counting them. Now, I did something to that original sample. I diluted it. So I have to remember that. And I have to include that dilution factor in my mathematical calculations. I didn't determine the number of colonies in the original sample. I diluted the original sample and then determined the number of colonies. So you have to do some math. You have to do some math so that you can report back an estimate, because this is an estimate, of how many cells there are in that original volume of material. But I can get a pretty good estimate by doing this kind of work. Now remember, and this is a really weird concept, I'm not actually counting cells, am I? I'm counting colonies. But a colony came from a cell. So the way we report this is we call them colony forming units. It's a cell that makes a colony, but we call them colony forming units. This unit of material, this cell, was placed on an agar plate and it was allowed to grow for 24 hours, creating a colony. I'm going to count the colonies. So on my report, it's not going to read, and I'm making this number up, it's not going to read 1 million cells per liter of lake water. It's going to say 1 million colony forming units per liter of lake water. Now, why don't we just call them cells? We're using the colony as a measure of cells, so why don't we just call them cells? Because it wouldn't be accurate to do that. It is possible when you're spreading cells out over a plate, it is possible for 2 or 3 cells to land on top of each other. And then the next day, when you're looking at that colony didn't come from 1 cell dividing. That colony came from those 3 cells dividing. So we don't call them cells. We can't be 100% sure that each colony came from 1 cell. So instead, we say colony forming units, or what we shorthand as CFU, colony forming unit. And I can do these spread plates and serial dilutions on any environmental sample that comes in. Sometimes I'm going to figure out CFUs per unit of square feet. Sometimes I'm going to find it per volume of air or per volume of water, whatever the original sample was that came into the laboratory. Okay. So Elaine's asking about the slides that are posted on Canvas for this lab. And you're correct, Elaine. There are a couple of slides in this, what I'm showing you here, some pictures that are not in that slide set. Generally speaking, if there's any video or there's a particularly large number of images that we go through in the laboratory, you won't see those in the slides up on Canvas. The slides that are up on Canvas are really more about the written things that are on those slides. So the reason I put those slides on Canvas, for these lab slides on Canvas for us, is in case you want to go back and look at written material. But if you want to go back and look at video, or you want to go back and look at all the images that we look at, you should go to the lab video. Okay, good point. Let me go back to 11 here. Somebody was asking about this slide. Remember, sometimes what I'll do for us is I will write some information down in this notes section. And that's so, again, if you want to go back, you can read some of the things I said in writing, just in case you're not sure how to spell it, or maybe I went over it really quickly. You can find this information sometimes in this notes section. And again, I just do that to try to be a help for us. So again, a serial dilution is a step-by-step dilution process, usually by a factor of 10. So I'm going to take my original sample and I'm going to dilute it 1 to 10. Then I'm going to take some of that and I'm going to dilute
Can't
it.
see the presentation. Sorry.
I'm sorry, you can't see what?
The presentation.
Sorry, my fault. Oh, I don't know why it's doing that. Hold on. Let me try to do this fresh. There, everything good now? Are you looking at the series of spread plates? Or I should say, anybody not seeing the series of spread plates? Sorry about that. Sometimes I forget to refresh the share. So down here in the notes section is just some of the things that I said about making these spread plates. Remember, it's called a serial dilution. When we do a series of 1 to 10 dilutions on our original sample, and then we make one plate for each dilution. Remember, we can use pour plates if we're concerned that the microbe maybe doesn't like to grow in oxygen. CFU is the unit that we use when we count colonies. Colony forming unit. It's an estimate of the number of cells in the sample. When we count up the cells, or the colonies, sorry, we want to count on the plate that has between 30 and 300 colonies. Just because we're going to get our best, most accurate count with that plate. We have to remember, though, to multiply by the dilution factor when we're doing our math. So if I made this plate, for example, with my 1 to 1,000 dilution, I have to include that in my math. I have to include the fact that I diluted this by a factor of 1 to 1,000. Good questions. So we're really doing two different things with environmental samples. Sometimes we're trying to quantify how many microbial cells are in a sample. We just want to know very generally how many microbial cells are in a particular environment. Other times when we're doing environmental samples, we want to know what microbe is growing. What is the genus? What is the species? So we have different techniques for those different questions. When we use a Rodak plate, we're not going to know the genus and species, but we're going to get a good idea of how many microbes are growing in that area. When we do a spread plate or a pour plate, we're not going to know the genus and the species. But again, we're going to quantify how many cells are in that environment. We can identify the microbe. We can, but we have to do different tests on it. We might make a quadrant streak plate, get some good colonies growing, and look at the colony morphology, for example. That's not going to tell us the species exactly, but it can give us some information, and we can do some additional testing to identify that microbe. So it all depends on what question we're asking about that environmental area. Sometimes all we need to know is how many cells are growing in this place. Other times we need to know what is growing. All right. So on this slide, we're looking at some of the things that I've said earlier. Sometimes our purpose is to determine the number of microbial cells in a particular environment. We have to remember that we're taking different kinds of samples. If I'm sampling a solid surface, I can come up with a quantity of cells, or at least a good estimate of how many cells are growing per surface area. If I'm testing the air or the water, I can do it per volume of that material. But I'm always going to report it as colony forming units, or CFUs. All a colony is, remember, is a mass of bacterial cells that I can see with my eyes. I don't need to go under the microscope. It's a visible mass of bacterial cells, and they are all clones. They're all genetically identical because they all came from one cell that I spread out onto that agar. The parent cell divides and divides and divides. You get 2 cells, then you get 4, then 8, then 16. That's exponential growth. It seems crazy, but 24 hours later, I can have millions of cells in one colony because they divide that quickly. When we count colonies and we report that as CFUs, that's an estimate of cell number in that original sample. All right. Any questions? Yes, there are not always notes under the PowerPoint slides. Sometimes I don't put any notes down because I don't think you need any additional notes. But if I feel like there's some additional notes that might be helpful to you, if you go back and take a look at the slides or take a look at the video, I'll put those in there. You wouldn't be able to pull those notes, move those notes, unless you pull up the slides. That's why I include the slides on Canvas. Sometimes, in other words, sometimes, depending on how we're viewing the slide, the notes will be visible and sometimes they won't. You have to move that notes box up and down in order to see them. So we're going to switch gears a little bit here. We're going to leave environmental sampling behind. Or actually, there's a couple other things I want to mention about environmental sampling before we move forward, before we move forward into a little bit of discussion of culture media. I wanted to say a couple things about this counting process. We're going to make a whole series of spread plates from that one sample. We're going to take that one sample and we're going to make dilutions of it. And then each dilution goes onto its own plate. And then I'm going to put all the plates in the incubator. And I'm going to come back 24 hours later and I'm going to choose the plate that has between 30 and 300 colonies on it. I can eyeball that. Now I'm going to count those colonies. How do I do that? It is possible to just sit down and literally count all the colonies on the plate. And if we're in a teaching lab, we do that with students. Or if we're in a lab where we don't do this very often, we do that with students. But if you're in a lab that counts a whole lot of plates every day, that can be really tedious. It could be very tedious to sit and look at this plate, for example, and have to count all these individual colonies. One of the tricks that we use is to take a Sharpie pen. Remember, there's a lid on this plate. And touch the Sharpie pen to the lid for each colony that we're counting so that we don't count them twice. But again, if you're counting a lot of plates, that can be very tedious. So we have some devices to help us. This is one of the devices right here. This is a semi-automated little piece of machinery. It's not expensive. This is a picture of someone using it down here. What you do is you place your plate right over this light illuminator. You can see there's a little grid visible in the illumination. That's going to help us divide the plate up into sections. This person is holding a stylus. And all she's going to do is she's going to touch the stylus to each colony. And the machine is going to keep count for her. See that? The machine, every time that stylus touches the plate, touches the plastic lid of the plate, the machine's going to keep track. Now, she doesn't have to count every single colony. One of the tricks we use is we divide the plate into 4 sections. Because these colonies are relatively evenly spread out, if we divide the plate into 4 even sections, she can count the colonies in one section and then multiply by 4 and get a pretty good estimate of the number of colonies on that plate. So you can do it by hand. You can do it with a semi-automated machine like this. And then the busiest labs, the labs that are doing lots and lots of spread plates every day, they're going to use a fully automated computer-based system to do all their counting. That's actually where this screenshot came from. If I enlarge it, you can see that we're on a computer screen here. What these fully automated systems do, you take the plate and you put it underneath a camera. The camera snaps an image of the plate, and then the software counts the colonies for you. Obviously, you have to buy that computer program. So some laboratories use that, some laboratories don't. It depends on how much counting they're doing every day. You can count by hand. You can do semi-automated counting, and you can do fully automated counting. The other thing I want to say another word or two about is the Rodak plates. Remember that the Rodak plates don't tell us what is growing. They just tell us how many. We're going to count up the colonies that grow on these Rodak plates. And because this is a commercially available system, it also comes with tables to tell us what that count means, like how we should interpret it. So if it's my job, part of my job in the hospital, to go into the operating room once a month and press a Rodak plate onto all the different surfaces in the operating room. I'm going to take those plates, I'm going to put them in the incubator. 24 hours later, I'm going to count the colonies on the Rodak plate. I can go to these tables that the company makes and sends to you with the kit. I can go to the tables and I can interpret the count. Let's take a look at what the general rules are. For each of my Rodak plates, if I get between 0 and 25 colonies growing on that plate, that's considered good. Remember, microbes are everywhere. I'm always going to get microbes on surfaces. I'm always going to get a couple of colonies on a Rodak plate. If I have a few number of colonies growing, it's not a problem. But if I have, let's say, 26 to 50 colonies on my plate, well, that's really a fair result. Maybe I need to step up my disinfection procedures a little more. If I get 50 and over, that's considered a poor result. That's a lot of cells growing on that one plate. Remember, I touch that plate to just one place. I press it down on one place, on one surface. If I'm getting a whole lot of colonies growing, I don't think my disinfection procedures are going very well. I need to change something at that point. So the Rodak company is going to send me a chart like that and tell me use these standards to tell you how well you're controlling microbes in your environment. But a lot of institutions, they don't use the Rodak chart. They make their own chart and they hold themselves to an even stricter standard. In other words, in a lot of hospitals, the way you interpret the Rodak plate, it's going to be different depending on where in the hospital you are. If you're in an operating room, you don't even want 25 colonies on your plate, right? If you're in a patient room and you're sampling, say, the bathroom floor, maybe a few extra colonies isn't as bad. It's going to depend where you are. Remember too, just like we said with the water, just like you have to get a representative sample of that swimming area, you've got to get a representative sampling of surfaces when you use Rodak plates. You can't go into a patient room and touch one plate to the tray table in the patient room and that's all you're sampling. You got to take several plates in there. You got to take 10 or 20 plates in there and get samples from all over the different areas of the patient room. And then you go to these charts. And the charts help you see how good you're doing controlling microbes in that environment. This is a chart, for example, that I found from a particular hospital system. This was their published chart that they used in their infection control procedures. As you can see in the first column, it lists different areas. So for example, the critical areas that they are monitoring, they're monitoring the floors in the operating rooms, in obstetrics, they're monitoring the floors in the nursery. They're monitoring tabletops around in the hospital. They're monitoring the floors and the tables in the patient rooms. They're monitoring bathroom floors and sinks and tubs and toilet seats, other floors, other surfaces, all over the hospital. And there are different needs in different places. And then what they've got are CFU counts and how they should be interpreted. So for example, if we go into the floor in the bathroom and we take a Rodak plate and we press it to the floor in one of the restrooms that is available to the public, let's say, we're going to tolerate more microbes on that surface than, say, the floor of a patient room. You see what I'm saying? There are different requirements in different places. So let's take a look. In that patient room, it's okay if I get up to 25 colonies on a Rodak plate from the floor, that's okay. I'm still doing a pretty good job at my disinfection procedures. But if I press a Rodak plate onto the table, the patient tray table, I'm only going to tolerate 5 colonies. 5 colonies, I'm still going to say that's a good result. If I press a Rodak plate to that patient table and I get 25 colonies growing, that's a poor result. Anything over 16 colonies for this particular hospital system is considered a poor result. I'm not doing a good enough job disinfecting surfaces in between patients. You can see that the strictest standards are in the most critical areas. the operating rooms, the nursery, the places where I really have to be sure that I don't have a lot of microbes because the risk is just too high in those places. I want to get it down between 0 and 5 colonies on my plate in order to be able to say I'm doing a good job disinfecting. So we have to sample our environments. We have to sample our environments because we need to know whether we're getting exposed to dangerous pathogens. We need to know whether we're doing a good job with our disinfection protocols. We need to know. And so we monitor. We can count colony forming units on spread plates quite accurately. And that can give us an estimate of how many microbial cells were in that sample. We can sample surfaces by just pressing a Rodak plate down or by taking a sterile swab and running it across a surface. We have to remember, though, that a swab across one surface or a Rodak plate pressed down on one surface is only telling me about that surface. I have to be careful that I don't use one sample as a measure for an entire facility or an entire collection of rooms. There are limitations, in other words, to our sampling procedures. And the limitations are I'm testing one particular place. I need to make sure that I'm getting enough samples so that I really have a representative look at what it is I'm trying to sample. All right. Questions. There are people who do this work in hospitals. There are people who, as part of their job procedures, their job description includes working on infection control. And their job is to monitor different rooms and different areas in the hospital in order to be sure that whatever's being done with the janitorial staff, whatever disinfection processes are being used, are effective. And of course, hospitals have a particular challenge in keeping pathogenic microbes at bay. As you probably know, there are some very dangerous microbes that tend to grow in those dark spaces inside hospitals. It's hard. It's hard to control microbes in a place where sick people gather. It's tough, and it requires a lot of monitoring. There are also people who have really, I think, very interesting jobs, for example, with the state of New Hampshire, who get to go out during the summer months and sample, bodies of water and take them back to the lab and do some testing. I mean, I think that'd be a fun job, but I'm a nerd, so. But yeah, from time to time in New Hampshire, you might go to your favorite lake or pond, and you may see a notice from DES, from the Department of Environmental Services, saying that it's not safe to swim right now, and that the beach is closed temporarily. And that's because they have taken a monitoring sample and they have found too many microbes right now. That happens quite a bit in New Hampshire because the places where we swim are natural bodies of water. They're not pools that are being treated with chlorine. They're natural bodies of water. And from time to time, the number of microbes just gets too high to be safe. So the state will close a beach for a few days until nature takes its course and the number of microbes drops back down. Connie's saying that she has some friends who are in water testing for Cape Cod. Oh, that sounds great. I would love to be on Cape Cod gathering water samples. That sounds like a lot of fun. Oh, One of the biggest problems we've had with natural water bodies in recent years has been a type of bacteria called cyanobacteria. You may have heard of this organism. We used to think that this was an algae. So we used to refer to this organism as blue-green algae. It's not an algae, it's a bacterium. It just so happens to be photosynthetic as well. So it's a bacterium that can do photosynthesis. Pretty amazing. When it overgrows in a body of water, it creates that green slime that you'll see sometimes. So around the edge, you'll see like a thick, slimy, like mat of material. A lot of people will look at it and go, oh, look, algae. But sometimes it's not algae. Sometimes it's cyanobacteria. And cyanobacteria actually creates toxins. And so if we detect a growth of cyanobacteria, we will close a beach, because those cells that have overgrown and created that mat of green, they're going to secrete toxin into that water, and that can really injure or even kill. So yeah, from time to time, you'll notice beaches closed in New Hampshire because of cyanobacteria. And of course, the reason that bacteria overgrow like that is because there's too many nutrients in the water. That usually comes from runoff. Lots of fertilizer flows into the water. It allows those cells to grow like crazy. And sometimes we get into trouble. But nature will take its course. Those cells will die off within a week or so, and then the water will be safe to go back into. So yeah, very interesting. Cyanobacteria has been a real problem in our state in recent years. Elaine's talking about being really careful. Yeah, well, anyone who is immune compromised, anyone who has chronic illness, anyone who, like Elaine is saying, has, say, a central line or a G-tube or a catheter, they have to be careful when they go into these natural bodies of water, because first of all, they've got something, they've got a physical entry point into their body that might be dangerous, but also because they're ill. They're fighting some sort of an illness, and their immune system is not quite as able to fight off some of the infections they might pick up in bodies of water. Generally speaking, like Elaine is saying, we recommend to people that if you've got an opening into your body with a medical device, that you cover it, with a transparent bandage, some kind of a sealing bandage before you go into a body of water. It's just safer. You wouldn't want to get even a mild infection around that. It wouldn't be worth the hassle. It's just safer to cover it before you go in. Yeah, and Connie's talking about the problems with the water table changing in places like Cape Cod, and also septic systems. Yeah, that's a great point. When you live in a place that's fairly rural, and a lot of New Hampshire and other parts of New England are fairly rural, the islands, of course, Cape Cod and Nantucket, they're not able to hook into municipal water supplies and municipal waste systems. So they're on septic, they're on wells, and they're on septic. And a well-run septic system is great. It handles dangerous microbes very well. But unfortunately, a lot of septic systems are not well maintained. A lot of residential septic systems are not well maintained. So if you get a change in the water table, you get more water into that sandy soil, for example, you can have a problem with septic systems being a source of bad bacteria. And those things have to be, you have to monitor. A place like Cape Cod, it's such a natural place in our minds, right? It's this lovely little, magical beach community. But the problem is, you got a lot of people living there. You got a lot of septic systems. It's a potential source of bad microbial growth that can contaminate other water sources. That's exactly right. There are parts of our country where the septic systems are so bad, so out of date, so badly managed that even the municipal water is dangerously contaminated. There are parts of the South, for example, where septic systems have been allowed to degrade to the point where they're not even functioning. And then you have human waste out in the environment that is not being managed, and natural bodies of water get contaminated. Yeah, it's very interesting. It's a very interesting area of microbiology. All right. So we're going to switch gears here. We're going to talk just a little bit about culture media. Now, we've got about 20 minutes left. We may not get all the way through this, but if we don't, we'll pick it up the next time we get together. I want to introduce some new vocabulary today about culture media. So far, we've been talking about how we use culture media in the laboratory to create, again, a growth of microbes. We are purposefully growing a population of microbes in the laboratory with this culture media. This media allows us to grow microbes that we've collected from the environment. It allows us to grow microbes we might have collected from a patient. We can grow microbes that we're using for research purposes. We can grow microbes that we want to use for industrial purposes. So culture media has to contain the things that microbes need to survive. And there are lots and lots of different kinds of culture media, but at its most basic, culture media has to contain 4 things. First of all, it's got to contain water. Every living thing on this planet needs water every day, so it must have water in it. It also has to have some kind of carbohydrate in it, and that's for energy. Now, in a couple of weeks, we're going to start talking about metabolism, and we're going to talk about where microbes get energy from. What we can say today is that the material, we'll call it dietary material or nutrient material that microbes are going to use for energy are carbohydrates. We also need to put in what we call a carbon source into the media. Now, generally speaking, this is protein. When I talk about a carbon source, I'm talking about a source of carbon that the microbe can use to build other cellular molecules. Remember, all life on this planet is carbon-based. It's organic. That's what that word means scientifically. All of our cells, all cells, are carbon-based. So all of our molecules use carbon as sort of the skeleton. And microbes will get that carbon from protein sources. Now, the other word I have here is peptone. We're going to talk a lot about peptone this summer. All peptone is pre-digested protein, or I should say partially digested protein. So sometimes in culture media, we put just plain old protein in there, just like you and I would eat protein. We put protein into the media for the microbes to eat. But sometimes we'll take that protein and we will break it apart a little bit. We'll sort of pre-digest it a little bit. That's what peptone is. It's protein that has been broken down a bit before we give it to the microbes. So it's more easy for them to metabolize. Now, the last thing that must go in to all culture media is a series of what we call, I wrote macro, it should be micronutrients. These are things like a source of nitrogen, a source of phosphorus, a source of sulfur. These are the little quantities of nutrients that all microbes require in order to meet their metabolic needs. Remember, when we use the word macronutrient, we're talking about protein, carbohydrate, and fat. Micronutrients are different. Organisms need those in very small quantities, but they need them. They have to be available to them. So there are lots and lots of culture media, more than we can possibly talk about even this summer. But we can group them. We can group all of the culture media according to how they're made and what they're used for. For example, one of the ways that we group culture media is whether or not it is chemically defined or what we call complex. So a chemically defined culture media is a medium that has only known ingredients. In other words, when we're making this media, we're putting in things that we know exactly what they are. Now, you might say to yourself, well, how would you not know what something was? And the answer is there are some natural materials that we will put into culture media sometimes that have an unknown chemical composition. For example, in complex media, media that has ingredients that are not 100% known, sometimes we use things like blood. We will use blood gathered, for example, from sheep, and we'll put that in the media. Sometimes we'll use egg. It might be egg yolk, it might be whole egg. Sometimes we put different kinds of milk products into culture media or meat extracts. Now, the reason that we put these kinds of complex ingredients into culture media is because some microbes have very particular nutritional requirements. Some microbes, just like some other organisms, have a very strict list of things that they must have in order to grow. And because we can't know exactly what they need, we give them nutrients that are very rich, very dense. Blood, as disgusting as it sounds perhaps to some of us, is a very nutritional liquid. It's a semi-solid, right? It's full of different types of protein, it's full of different lipids, it's full of different carbohydrates. We can't make a list of exactly what is in blood. We can't do it. Just like we can't tell you every single molecule that's inside some of the other complex ingredients. So there are little key molecules in these undefined ingredients that some microbes just need in order to grow well in the laboratory. Some microbes are just so easy to grow. All you have to give them is a little bit of protein, a little bit of sugar, and some water, and they'll grow great for you. But other microbes are very, very particular in their dietary needs. And so we add in these more mysterious ingredients. So that's what we mean when we talk about a complex media. It's got some kind of an ingredient in it that it's a natural product, but it's not well defined in terms of the actual molecules that are in it. There are chemically defined media. We know exactly what's in them. And then there are complex media that are a little more mysterious. Now, there are some media, both chemically defined and complex media, that are just for growing purposes. We just want to grow the microbe. That's all we care about. So, for example, we have several really good chemically defined media that are just general purpose. For example, there's something called nutrient media. It comes in a liquid form and it comes in a solid form. So nutrient broth, that's the liquid, and nutrient agar, that's the solid. It's chemically defined. It has a known list of ingredients, or I should say a list of known ingredients. It's not a very, difficult media to make. And most organisms will grow for us in that media. There are some that won't, but most will. Now, when I use this word agar, agar is a carbohydrate. And if you remember from lecture, agar is extracted from algae. It's part of certain species of algae. It's part of their cell wall. It's a carbohydrate. The types of algae that make agar in their cell wall are red algae. That's the category of algae. Now, agar makes a great material if you want to have a solid culture media. Agar is a lot like gelatin. It has the ability, when you mix it with water, to solidify, but not get hard. I've mentioned before in here that if you took an agar plate and you took your inoculating loop, if you weren't careful, you could push your loop right into the agar because it has the consistency of gelatin. If you've ever had Jello gelatin, most of us have, it has a very similar consistency to that. So it's solid, but it's soft. Now, the agar is the carbohydrate that creates that gelatinous material. We add other ingredients to the agar. So the agar is just sort of the matrix of that solid material. We add in carbohydrate, we add in protein, we add in micronutrients. The agar is just what holds it together. Now, it's a great material for a couple of reasons. Probably the most important reason is this, number 3. Microbes can't eat agar. They can't digest it. They can't use it as a nutrient. They will grow on it, but they can't degrade it. If you think about it, we don't want to grow microbes on something that they're going to consume. We need that solid surface to stay solid, in other words. So it's very easy to add nutrients to agar. The agar is just the matrix that holds the media together. The other nice thing about agar is it's going to remain solid in the incubator for us. It doesn't liquefy until a very high temperature. You have to get it up to about 100 degrees centigrade. That's when it will melt. But in a regular incubator, which is typically held at about 35 to 37 degrees centigrade, the agar is going to stay solid. We liquefy it when we're making it and we pour it into plates. It's super easy to work with. And it gives us that nice, smooth, solid surface to grow colonies on. It's a great material. And here's the thing, you can buy agar commercially. You can buy a powdered form of agar. And then all you have to do is mix it up and liquefy it in the microwave and pour it into your plates. And that's what you're looking at in this picture. You can also purchase pre-made agar plates, but most laboratories make their own. They buy the agar powder and then they add in whatever ingredients they want to add in, whatever nutrients they want to add in, depending on what kind of bacteria they're trying to grow. So you can pour agar into a petri dish and let it solidify. That's what you're looking at in this image here on the top right. But you don't have to use agar in a dish. You can also pour agar into test tubes and let it solidify. And we use these tubes for different reasons. Generally speaking, if we're using agar in a petri dish, it's because we're either going to do a quadrant streak or we're going to do a spread plate. But sometimes we don't need to do either of those things. Sometimes we need to grow some bacteria, but we don't need to do a quadrant streak or a spread plate. So you can pour a little bit less agar into test tubes and use it for other reasons. Now, there are two kinds of these agar tubes that I want us to be familiar with. One of them is called a slant, and one of them is called a deep. Let's take a look at those. You can pour agar into a test tube, and if you leave the tube at a slant, at an angle, while the agar is solidifying, it will solidify like this. It'll solidify at an angle. And what it creates is this oval of flat surface that we can spread cells on. Now, generally, the reason we would use a slant is because we're doing some kind of metabolic test. There's something in the agar that is specifically going to test this microbe's ability to perform some kind of metabolic pathway. So we smear cells on this slanted surface, but we don't need to grow colonies. We don't need to do colony morphology. We don't need to count anything. We just need cells to grow. And so we can spread them out on this oval surface and they'll grow for us in basically a smear. Now, other times, again, we will work with organisms that don't like oxygen. Some organisms even find oxygen toxic. So if we want to grow those, we use what's called a deep. This time we pour agar into the test tube and we keep the tube upright as the agar solidifies, and you basically just get a tube full of agar, solid agar. What you can do with a deep is you can take your inoculating loop and you can cut off the loop. You literally cut the loop off of the end of that wire, and you're left with what we call an inoculating wire. You can dip that wire into your sample or your culture, and then you can press the wire down. You can sort of stab the agar with that wire. If you look down here on the bottom left, these are two agar deeps that have been inoculated. This looks like it might be broth, but it's not. It's agar. It's that gelatinous material. You can see a white line here and another one over here. So I've taken my inoculating loop, I've cut the loop off, so now I just have a wire. I've dipped that wire in cells, and then I've stabbed those cells down into the agar. If these are cells that don't like oxygen, they're going to grow for me down here because this is not going to have a lot of oxygen in it. Sometimes they just grow right in that stab line. That's what you're looking at here. But sometimes this happens. This is interesting. Notice that over here, the line has gotten much wider and it's kind of fuzzy looking. What's growing in here is mobile. It's motile. It has flagella. And the microbe is leaving that stab line and actually swimming out into that agar. It's actually pushing its way through that gelatinous material into the agar. So that's what a deep looks like when we grow organisms in a deep. And again, we would do that because we have reason to believe that this organism doesn't like to be around oxygen. All right. The last couple of slides in the slide set are a few more words about colony morphology. And we're not going to have the time to go through them today, but the slides are there in the slide set for you. And I'm going to post for us a short video on our playlist that walks you through those slides. Remember, each bacteria grows in a particular type of colony. Each species of bacteria grows in the same way every single time. It's going to have a particular color, it's going to have a particular shape, it's going to have a particular size. That's what's called its colony morphology. Some bacteria have very unique colony morphologies. In fact, there are some species where you can look at the colony and you can already know what you have. Very few, though. Sometimes two different bacteria can have the exact same colony morphology. So you cannot look at a plate, look at the colonies growing, and know for sure what species you have. But it does give you information. It gives you a piece of information about whatever it is. Just like gram staining gives us a piece of information, colony morphology gives us another piece of information. And the more information we can gather in the laboratory, the better able we're going to be to identify that species of bacteria. So when you have a few minutes, go ahead and view the colony morphology supplement video that's going to be up on our playlist. And if you have any questions about what you see in that video, of course, you can message me or bring those questions to lab next time. All right. So remember, you have a lecture exam to complete today, if you haven't already. That needs to be submitted by 8 o'clock tonight. Don't forget. You're ready now to do your lab homework. That will be due by Sunday at midnight. And I will see you again on Monday. All right, everybody, enjoy the day. Should be a nice sunny day for us. We'll see you next time.